Today I'm going to take you through an exam paper and show you how I decipher questions and find clues in questions that will help me have an idea as to what to put in the answer to get marks. We're then going to mark my imperfect answers. I know the biology, but I haven't learned this mark scheme and I will not get every mark. And I'm going to talk to you about how I analyse my answers to try and learn from my mistakes, but also talk to you about some marks which I consider to be written off marks, marks that I would never ever have thought of writing. So differentiating between those marks that I feel like I should have got and those marks that actually I know I would never have thought of writing. I'm just not going to worry about them. So let's start on the first question. So you're given two diagrams, one with or both with chromosomes, one of them arranged in pairs. And you're asked to give two pieces of evidence from figure 1 and to explain your answers. So each answer requires two bits to it, the evidence and a biological explanation, i.e. what is going on behind the piece of evidence that you've thought of. So here are my answers. Chromosomes are visible, therefore, that is because DNA has condensed. Chromosomes are only visible in the cell just before a cell division. So the fact that we can see chromosomes tells you that some kind of cell division is happening. Now, it says undergoing mitosis, and I looked at this picture and nothing really screamed at me about why it was mitosis rather than meiosis. So the other answer that I put was that, the chromosomes have two chromatids joined at a centromere. I added this bit just to make sure I was getting my terminology in, and that is because DNA has replicated. Looking at the mark scheme, I got mark point 1 and I got mark point 2. Mark point 3, I understand what the examiner is getting at, that the chromosomes weren't arranged in homologous pairs, but I kind of know that that happens partway through prophase, so it wasn't an obvious piece of evidence to me, but fully understand why other students would have thought of that. Next question, tick one box that gives the name of the stage of mitosis. Well, by a quick process of elimination. Anaphase is when the chromosomes are like this in the cell. Interphase is when all the DNA is completely uncoiled. There aren't even chromosomes, so it's definitely not interphase. And telophase is when you've got either the chromosomes or actually nuclei at each pole of the cell, so it's not telophase. And prophase was the right answer. When preparing cells for observation, they put them in a solution that had a slightly higher water potential. As soon as I see this word water potential, I'm thinking osmosis, because water potential and osmosis absolutely go together as terminology. So suggest how this procedure moved the chromosomes apart. I talk about osmosis, and because I talk about osmosis, I put water potential in my answer. Again, because those terms go together. This increased water volume moved the chromosomes apart. So what was on the mark scheme? Yep, I did get that mark right. So I got mark point 1 here. Ignore references to changing water contents of the cell. So I would not have got this mark. So according to the way that I analyse exam answers, which I have outlined in my video, Improving Your Grade to an A star, I would say that I have a communication terminology issue here, or I'm going to say it's a write-off mark. I'm not sure I would have thought of writing it in quite the way that the mark scheme wanted it. So I'm not very worried about this mark. I'm just going to put write-off question mark. Notice how the simple annotation just on the side of the exam is going to help me later on. This is not rocket science analysis. Next question 1.4. The stain binds to some areas of the chromosome more than others. Suggest one way the structure of the chromosome could differ to cause that. This is not an obvious answer to me. So I'm kind of going, how does the structure differ? And the only thing I can think of, really, well, there are two things that I could think of, either base sequences or the level of binding to histones. I've gone with one rather than the other because notice they've asked for one way. If I wrote two down and one of those was wrong, it would cancel out my correct answer. If it says one, only write one. So mark scheme does say differences in base sequence, so I've got the mark there. Here, figure 2, the chromosomes are arranged in homologous pairs. Okay, so what is a pair, a homologous pair of chromosomes? And that was what got me the mark, this bit here, which I can understand. And here give two ways in which the arrangement of prokaryotic is different from the arrangement of human DNA. So this is interesting because it's the arrangement. It actually isn't the structure of the DNA, it's the arrangement of the DNA, and that gives me, I noticed that wording, and that gave me more scope to write things as answers. So I wrote that the prokaryotic DNA is circular, whereas the eukaryotic DNA is linear, and prokaryotic DNA is not coiled around histones, whereas human DNA is. According to the answer, I got mark point 1 and not associated with histones. I got that mark point as well. You could have also talked about the fact that there were plasmids in prokaryotes as well. That gave me the 2 marks. As I read this question, I drew myself diagrams to understand what was going on. So I worked out that there were cubes of potato and each cube was 35 millimeters. The sides were 35 millimeters, but he then cut another cube into 8 equal size smaller bits. And I just kind of went, how would that work? Well, if he cut it this way, that would be 2. If he cut it that way, well, that wouldn't be 4. Hold on. So if he does 3 cuts, he's going to end up with 3 smaller cubes, each of which have got a side of 17.5. And he put them into equal concentrations of sucrose and left them and weighed them. And this is what he got. So describe the method the student would have used to obtain the results in figure 3. So to obtain the results in figure 3, it is worth looking carefully when they give you questions like this. There are clues in figure 3. Start after all the cubes have been cut and consider control variables. Pat cubes dry thoroughly, I thought. Weigh and record mass of the single cube and all the smaller cubes. Place them into the sucrose. Put bungs in to stop evaporation. It's one of those details of this particular practical, which I'm going to be honest, I never really understood how much water is going to evaporate in the time that this practical goes on. Anyway, let's move on. Right, here I started going, okay, let's have a look. So here am I trying to see whether there were obvious times at which the cubes were weighed. Now, it seems to me that possibly 40 was one, but it isn't really obvious. So I guess that I would weigh them every 20 minutes. But look, it actually tells you how long the experiment went on for. So every 20 minutes, remove the cubes, pat them dry, re-weigh them, 8 cubes together. Yes, continue until 160 minutes. I picked up the clue from the question, and then I went back and went, oh, I haven't done my control variables. What control variables can I think of? So I thought of the concentration of sucrose solution was kept the same, the species of potato, the time intervals for recording mass, and the temperature. It was all I could think of. Right, so I didn't think of that one. I probably should have remembered it because I absolutely know that part of this practical is about shaking the cubes regularly to mix them around in the solution. Method of controlling temperature, I didn't get it. Method of drying cubes, I got that one, and measure mass at stated intervals. So I got 2 out of 3. And I'm going to be honest, I think an awful lot of students probably only got 1 or 2 marks out of 3. I think those were hard marks to get. Here you had to work out the rate of osmosis between 0 and 40 minutes. Is it different? And use calculations. Okay, calculations means do sums with numbers. So we had to work out the rate of osmosis between 0 and 40 minutes for both of these sets of cubes. So they both start off at 62. Be careful of the scale on axes when you're doing this kind of thing. And at 40 minutes, B had ended up at 53 and A had ended up at 57. So the difference for A is 5 and the difference for B is 9. So A, the mass loss in 40 minutes was 5. To make it into a rate, it's 5 divided by the time. Any rate is something divided by time. And that gave you a rate of this 0.125 grams per minute. That's the loss, the rate of loss. But the area of the cube, you then had to take into account that it was per millimetre squared. Now, per millimetre squared, that's area. It's not volume. Volume would be millimetre cubed. It's per area. So we have to calculate the area of cube A, which I have worked out. The area of a cube is the area of one face, which is 35 times 35, because it's 35 millimetres long. But there are 6 faces on a cube, so it's 35 squared times 6, which ends up being this rather frighteningly big number, which means that the rate is absolutely tiny, which is this. Notice that these minus indices just means that the grams, these tiny amount of grams, have been divided by the number of minutes. It's per 1 minute and it's per 1 millimetre squared. Whereas B, the rate of loss was this. If we did the same scenario, now notice the rate of loss in B is the size of the area of the cubes, but it's times 8 because of course there were 8 smaller cubes. And this ends up showing that B is actually slightly slower than A, but you had to do the calculation to know it. I really didn't like this question very much. Okay, there are many species of bee. They tell you what bees are because they don't assume that you know about organisms. It's why they describe them to you. And they tell you that there are many species because otherwise it wouldn't make sense for them to really talk about the biodiversity of bees. What is meant by species richness? This is a definition. It's the number of different species present in a habitat at a particular time. I know that that is right. And here we go. Number of different species in a community. But notice that habitat is allowed. I think this is a really horrible question. From the data in figure 4, he made the 2 conclusions. Do the data support these conclusions? Do they support? It means yes or no. And explain your answer. Now, it's likely that I'm going to need to put some kind of biology in the answer. So the natural habitat is most favoured. So I'm looking back at this graph and I'm saying, what about these graphs says that the natural habitat, this one here, is most favourable? So to me, the most obvious piece of evidence is that the mean bee species richness is higher than the other 2 habitats throughout the whole time that it is being measured. And here, the mean number of bees collected is larger than the other 2 at between 50 and 200. But is there anything here that tells me it isn't the best environment? Well, only this bit here, so kind of between 200 and 250, but it doesn't drop below both of them. It drops below the farmland and then below the town. So I'm going to try and capture that. This is me trying to explain it using biological reasons why. I've made my point about bee species and I made my point about the mean number of bees collected. Now, I've drawn a line here on my answer because it was only towards the end of me writing my answer that I looked back at the question and realised that it wasn't how do they support, it was do they support. So I suddenly realised I needed to put a no part of the answer in. So I put this, but this figure, the mean number of bees, is lower than farmland from 200 days and lower than farmland and town from about 220 days. Let's mark that bit first. So I have a choice of these 4 marks. So I didn't say that, I didn't talk about the peak, I kind of noticed it, I wasn't quite sure how to word that, but I've got that one. Oh, lowest mean number of bees, I've also got that one. And the mean number of bees was, oh, and I've actually got that one, I'm quite surprised. So now let's look at this. The town is least favourable for bees, town is least, so again you're looking at evidence that is saying that the town is least favourable. The town are the spots, so we're looking for where the town is below the others. So I'm kind of thinking here and here and here and here. Oh, actually, no, not between, it's between there and there. Between 150 and about 210, 215, it's the lowest, has the lowest mean number of bees collected. And from about day 125 maybe up to 250, the mean bee species is lowest. Right, here I wouldn't have got this mark because the number that I quoted wasn't 125, it was 130. I'd obviously estimated that wrong from the graph. If I'm honest, I'm not quite sure where the explanations, I hadn't thought of those at all. So I think I've got 3 out of 4. what was the one consideration to make sure? So they had to make sure that the bees weren't damaged or injured, and that allowed them to identify them accurately. Well, to identify them accurately, they had to have some method of identification, and the most obvious one to me is that they had photographs as the clearest way. And I didn't quite know how to extend this answer, but I gave it a good shot. So I would have got shouldn't harm the bees, and to use a key. Oh, I didn't say that. However, they allowed use photographs, so I would have got the 2 marks there. This question says, suggest and explain. You have got to suggest something and explain the biology behind it, how they could have improved the method used for data collection. So I said increased number of sites and number of times sites were sampled. Now this is me, I'm going to be honest, this is me being a bit naughty because they've said 2 ways. And I just don't know whether this will have been included as 1 way or would be counted as 2. So I actually know that as I am writing this, I am taking a risk that I might knock out one of these marks. If this is 2 marks, I might be knocking 1 out by putting a 3rd option which isn't on the mark scheme. So I'm aware of that. I decided, for the purposes of this paper, to take the risk. And this is me trying to explain why I'm doing it. Right, I wouldn't have got that because I didn't talk about the line of best fit. I wouldn't have got that mark. Collected from more sites to increase the accuracy. Because I did write 3 and I only got 1 correct answer, I am actually going to play it safe and say that I didn't get a mark here. So I'm going to give myself a read the question response so that I absolutely keep to only talking about 2 answers. Next question. How do these names suggest, what do these names suggest about the evolutionary relationships? So I'm focusing in on this term, evolutionary relationships, and I'm thinking the one thing I want to talk about is how close their common ancestors is, which is what I went for. They've got the most recent common ancestor. And then I went on to say that this one is the least closely related because it has a more distant common ancestor. When I saw the mark scheme, I could have kicked myself because it was ridiculous that I didn't get this mark, because there was an absolute clue in the question. What do the names tell you about their evolutionary relationship? I didn't get these 2 marks, even though as soon as I saw the mark scheme, I thought, oh my goodness, that was by far the most obvious thing to say. However, I did get mark 0.1 for talking about the common ancestor. So this to me is a read the questions. I read the question, but I missed this clue. This is asking you why an enzyme substrate complex increases the rate of the reaction. What is it about the enzyme substrate complex? According to I was thinking about the induced fit hypothesis, and as I was writing, I'm thinking I'm going to put that term in. I'm going to make sure that I talk about the active site of the enzyme, not just the enzyme itself, and that I need to talk about either the tertiary structure of the enzyme's active site or the shape of the enzyme's active site, always talk about either the tertiary structure or the shape of the active site, and never just the tertiary structure of the enzyme. That is always my advice. And so I said that it changes slightly on binding to the substrate. Now, I know, and I teach this, that it is because that it puts pressure on bonds that makes them easier to break. And this is what lowers the activation energy of the reaction. Notice it's the activation energy of the reaction, it's not the activation energy of the enzyme. And I also wanted to put in this term here, that it acts as a catalyst. This bit here doesn't directly answer this question, so even as I was writing it, I was thinking that I was likely to be wasting time. Reduces activation energy, I talked about, due to bending bonds. Calculate by how many times. You are literally seeing how much this tiny number goes into 578. So rightly or wrongly, I transformed them into standard form. And if you are dividing two numbers by standard form, you take away this number from this number. So it is 2 minus 9. And two minuses, I was always taught, two minuses make a plus, so 2 plus 9 equals 11, which is where this number comes from. So now I am just dividing this by this, and that gives me my answer of 1.93 times 10 to the 11, which I know is correct, so I'm going to give myself the 2 marks. So, having a look at this question, so this is with Lactose and this is without. So here you're asked to calculate the rate of reaction with no Lactose at 2.5 as a percentage of the maximum rate shown. Now, I thought this was going to be really horrible until I realised that this y-axis is actually the rate of the reaction. So this is the maximum rate of the reaction for Lactose, didn't get any higher, whereas this is the rate of reaction for without Lactose at 2.5 here. So I think that it is 1.4 and 0.44, and that's because each tiny square there's 5 squares, and there are in effect there's 0.2 of a unit, so each tiny square is 0.04 units. So hence, as a percentage, percentages are just posh fractions. So it's 0.44 divided by 1.4. That is the fraction that you had to work out. And to make it into the posh fraction that is a percentage, you times that fraction by 100. So 0.44 divided by 1.4 times by 100 is 31.4. And I do know that that is the answer. Suggest a reason for the difference in the results shown in figure 5. So you had this kind of result, and this is rate of reaction. At all substrate concentrations, the rate of reaction was higher with Lactose. Now that says to me that Lactose had some action on the shape of the active site, which meant that it bound more easily with the substrate and therefore formed more enzyme substrate complexes. So I want to get some of that terminology into this answer. And I knew it had to bind to the enzyme somewhere other than the active site, because if it bound at the active site, it would have stopped the reaction, not sped it up. So Lactose bound to the enzyme somewhere other than the active site. And this causes the shape of the active site. Notice how I'm careful. It doesn't cause the shape of the enzyme. It causes the shape of the active site to change shape, which causes it to bind to ATP. And I've written this because I'm now making my answer specific to the question, the information that they've given me, forming more enzyme substrate complexes. So let's have a look at the answer. Oh, well, that's ironic. They've actually allowed that as an answer. But look, it causes the active site underlined word. You had to have it in to change shape, which meant more enzyme substrate complexes. I would only have got 2 marks. And again, this would have been a communication terminology answer that I wasn't putting in the terminology. However, I don't think that I would have written that, if I'm honest. Right, I deliberately haven't drawn this because I do know the general structure of an amino acid, but I wanted to show you how I remember how to draw it. So I always draw my central carbon atom with its 4 bonds. And then I remember that this is an amino acid. So it has to have an amine group at one end, and it has to have an acid group at the other. And I know that this is COOH. And that avoids me missing out this carbon atom. And these two are relatively easy because this one is an H and this one is the R group. Right, it shows RNA codons and the amino acids coded for each codon. Codons are the base triplets that you find in mRNA. And it gives you some properties. Oh, okay, all right. Positive, negative charge, okay. And you should have come across this kind of table in your syllabus. So this is saying that U on mRNA codes for, this is PHE, which is phenylalanine, whereas this is the correct answer because there are 3 bases for every amino acid, and they've told you the number of amino acids. So that is the right little bit of maths. Use the information that would, in DNA, there you go, that would result in a change from val to alla. This is where you have to remember, so we're looking at from this, so it would have been GU any letter to GC any letter, but that's on the mRNA. So on the DNA, it would have been CA to any letter to CG any letter. So actually, this is the one base change that is required. This one was a possibility, but actually this changed too, so that would have been, and it was a single base substitution, so therefore that is the right answer. This question is about the effect of changing amino acids in an enzyme. Use all the information and your knowledge of protein structure. There's a big clue. You're going to be talking about something about primary, secondary, tertiary structure and suggest reasons for the differences. So the most obvious one, that amino acid position 300 was not in the active site, and therefore now here I am using all of the information. So here the table shows us that glutamine is negatively charged and lysine is positively charged, and therefore that would have an effect on only one type of bonding, and that would be ionic bonding. There are 2 ways in which these amino acids could have affected the rate of reaction of the enzyme. they are either involved in the active site or they are involved in the tertiary structure of the enzyme, where a bond holds two bits of the primary structure of the enzyme together, which is what I have tried to talk about in this answer. So I would have got this mark and I would have got this mark, even though I also put this in as well. And I would have got that mark as well. Here is question 6. I'm just going to let you have a quick read of that. So the chromosome content, I simply copied what was in the diagram above. And after the second division of meiosis, I know that it is the chromatids that separate. So therefore, that is the correct answer. This question is describing the selective breeding of clover. So the first part of the question is filling in these boxes. So I would do that by using these numbers here and saying that 50 out of 54 didn't produce 2N gametes and 4 out of 54 did. So that's the proportion that I'm working it with, but we didn't have 54 plants, we had 56, 14 plus 42. 50 divided by 54 times by 56 gives you a number of 52 here and therefore 4 here. And I know that that is right, and that would have given me the 2 marks here. Scientists tested their null hypothesis using chi-squared statistical test. Okay, the calculated value. This is the value that you look up in the table. This was the number that they got at the end of their statistical test. And the critical value, so the value in the table at p equals 0.05 is this. This is clearly a much bigger number than this. P equals 0.05, that is the probability that this number is due to chance. So I've said that 350 is much greater than 3.841, so I reject the null hypothesis, and there is a much lower than 5% probability that the difference, so here am I, I'm not just saying the result is due to chance, I'm saying what the test was that they did, and this was a chi-squared test, and therefore it's the difference between observed and expected. And I then go on to say that the difference is highly statistically significant. So I got that mark. This is a standard phrase in a mark scheme because P is the probability that, and I'm going to put result because I can't, it depends what the statistical test is. Here it was the difference between observed and expected is due to chance. I knew that those 2 words were going to be in the mark scheme. I did say all of that and I didn't say that. So I did get 2 marks out of the, I did get both of those marks. Use your knowledge of directional selection to explain the results in table 3. Well, this is all because that selective breeding was used. Normally we talk about directional selection in natural selection, where in the environment selects the favourable alleles that are going to get passed down from generation to generation because those individuals with the favourable alleles survive and reproduce. Well, in this situation of selected breeding, it isn't natural selection that's doing the choosing, it's people doing the choosing. So I've just stated that it is genetically controlled and it's by a particular allele, because I'm going to be talking about alleles because this is selection. This is a really important term to use. The selective breeding selected these offspring to survive and reproduce. I'm basically adapting my natural selection answer and pass on these favourable alleles to the next generation. I don't know quite what's happened to the G there. And therefore the proportion of 2N gametes increased as the frequency of the 2N allele increased in the population. So let's have a look and see what I would have got. Oh, I think I said that. And they pass their alleles down and the frequency. This is an important mark point because, well, natural selection can be defined at A level as the frequency, or selection rather, any form of selection can be defined as when the frequency of alleles change in the population. So this, I remembered this at the last moment as I was writing my answer and therefore put it in. Okay, new question. We're now on to antibodies. Explain how it works. There's two parts to this question: how it works and why it's essential to use passive rather than active. So the first part of the question is how it works. So I said that it binds with the snake toxin, making it unable to bind to cells and causing damage. In other words, it neutralizes the toxin. And passive immunity is essential as it takes immediate effect. Active immunity would take too long because active immunity requires the body to make its own antibodies. So, mark scheme. Here it binds to the toxin and causes its destruction. I know I wouldn't have written that. I've read elsewhere that it stops it binding. However, I understand why this is a mark point, because you learn at A level that this is how antibodies destroy pathogens. They bind to the antigen on the pathogen and bring about the destruction of the pathogen. So I got that mark point, but I didn't get that mark point. And notice they wanted you to ignore neutralizing the toxin. This is not, they do not deem this an A-level answer. So I got 1 mark, but I know that I would not have necessarily written the other mark. I'm going to write off that other mark. Then they show you a procedure for collecting the antivenom. Y is a mixture from several snakes to contain several different toxins, variants of the venom, so that antibodies are made that are complementary, because I got to the end of this sentence and felt like I hadn't nailed the answer. And then I suddenly remembered that I'm talking about antibodies. I should use this term in my answer. Oh, I actually get that mark there. Different antibodies. I certainly got this one because it was that slight moment of genius that I needed. Yeah, see, I didn't say different antibodies. I don't know whether I'd have got that previous mark, but I would have got 1 mark out of 2. Oh, this is about the horses and rabbits producing the antivenom using only this information. In other words, this information here. Which animal would be better? Now, I'm hoping that you'd realise that a larger animal would be able to donate more blood and therefore be able to produce more antibodies. And it says use a calculation to support your answer, and I have done that. And I know that more blood is one of the answers, and then this is the other answer. So I did get the 2 marks. Here we go. More more antibodies collected. Did I write that? Therefore, more antibodies. Yes, I did. Why would they undergo observation by a vet? Well, vets care for animals, so it's to check that they're not being harmed. And I do know that that is the mark on the mark scheme. During vaccination, each animal is initially injected with a small amount and then with a larger amount. Use your knowledge of the humoral immune response. So we are now talking T cells, B cells, to explain this vaccination program. And the second one makes me think about the whole memory cell response. So there is my answer, and I've included about it binds to receptors on T cells. They then clonally expand by mitosis. They bind to specific B cells. How that happens. Didn't really know how much detail I was going to need to get marks here. Second injection causes much greater and faster production of antibodies by stimulating memory cells to change into plasma cells and produce antibodies. So I got that mark. So notice again that the mark scheme here is all about the B cells, and T cells are hardly ever referenced in mark schemes. I put them in. I would always say put them in just in case, but it's worth noting that the answers always focus on the B cells. Second dose produce antibodies in higher concentration. So I got that mark. I don't have to have said what was in the brackets. Okay, so we have this question here. This data. And they concluded the heat treatment damaged the phloem. So explain how the results support this conclusion, that heat treatment damaged the phloem. Now, I have to say, I suspect, I can't quite remember what's in the mark scheme, I suspect that the mark scheme talks about the biology of what happens in the phloem, rather than just saying, oh, this is the evidence in the results that support this conclusion. So I want to talk about what I noticed from the results, but also expand on the biology. So this is me talking about the results, and this is me talking about what happens in the phloem. But it's 2 marks. I wasn't quite sure what they wanted. Even though I know that, I didn't write it. I didn't get any marks from that because I didn't compare it to the control. I think the lesson here is when you see this word explain, talk about the biology behind the results as well as explaining how the results support the conclusion. Okay, scientists also concluded this heat treatment did not affect the xylem. Right, explain how the results in the table support this conclusion. Now again, because the difference in water content not statistically significant as the standard deviations overlapped. Now, already I'm realizing I have not talked about the biology here. So that's interesting. I get the first mark. There is the evidence that supports it, but I didn't talk about the biology, so I didn't get the second mark. My learning from that is that I need to read the question that explain equals talk biology, as well as the results in this case. Here's the final question. And these are the really hard questions at the end of the paper. It's worth you knowing that the difficulty of the paper as it goes from question 1 to question 8. So if this is difficulty, it kind of starts off like this. So at the end, it gets more difficult in each question and then drops back down, and it gradually ramps up until the most difficult questions are at the end. So worth you recognising that. These are hard questions. What would you conclude? Right, conclude. Don't be afraid to state the obvious. Yeah, if one thing is bigger than another. I would also be considering, have stats been done? So what can you conclude? It's only what the results tell you. And point out everything the results tell you. But look out to see whether stats have been done, because if they haven't been done, there is less that can be concluded. So here I would have got that mark. Yeah, there's 1 mark there. There's 1 mark there. No statistical tests. Have they got something about no statistical? Here we go. So I'd have got 3 for that mark. I think I might get that mark. I'm not sure. I'm not sure. 2 named enzymes. Well, this to me is pretty short and sweet. DNA helicase unzips the double-stranded DNA by breaking the hydrogen bonds to form 2 template strands. This is 3 marks, and I'm kind of going, okay, where exactly are the marks here? DNA polymerase, this is a really common misconception. DNA polymerase does not rejoin the hydrogen bonds that have been broken by DNA helicase. It forms the sugar phosphate backbone by bonding deoxyribose. So here am I realizing that I'm about to talk about the sugar and the phosphate, and we are talking DNA. So let's actually talk about the sugar that I know it is, deoxyribose and phosphate, together by a phosphodiester bond. I'm working hard to get those marks, but I can't really say anything else about those enzymes. So I'd have got that mark, joins the nucleotides and forms phosphodiester bonds. Notice, reject here. DNA polymerase forms the hydrogen bonds. It doesn't. It's a really common misconception. Here we go. This protein is thought to be involved with the binding of one of the enzymes required at the start of DNA replication. So it's the binding of one of the enzymes to the DNA. Because otherwise, the enzymes that start DNA replication couldn't do anything if they didn't bind to the DNA. I've suggested that it stops cyclin A binding to DNA. Now, can I say, I think my English is appalling here. So these results seem to indicate that when the antibody binds to cyclin A, it stops cyclin A helping the binding of the enzyme at the start of DNA replication, which is why the percentage of cells replicating goes down so dramatically. If there is no cyclin A, then that also causes a very similar reduction in DNA replication because the cyclin A is not there to help that enzyme bind. And here, RNA that prevents translation of cyclin A, but they've also added the cyclin A, this takes it back up to the original amount, the original percentage. Right, and on this question, I think I would have only got 1 mark. And here, this would be my application and drawing conclusions from data. So this would tell me that I need to practice more explanations and drawing conclusions from data that is given, particularly these harder questions. And finally, we come to the last set of questions. These are really useful to know that they're here because they're fundamentally questions that ask you about processes and are relatively easy. They're not so much application and drawing conclusions. So always good to try and make sure that you've got the time to do these. So again, two parts to the question: the growth structure, the overall structure, and how we breathe in and out. They want the whole lot for 6 marks. So I've pre-marked this with the exam points numbered exactly where I think I have gained the marks. Looking at the mark scheme, I think I have lost 2 out of the possible 6 marks, and that is because of terminology. I lost this mark here because I didn't use the term thoracic cavity. Look, they particularly want you to use that term. And again here, I didn't talk about the internal intercostal muscles contracting. I simply talked about the external ones relaxing. So lost these 2 marks. This was frustrating, and therefore I'm definitely making a note of that. This is asking you to compare and contrast the structure and properties of triglycerides and phospholipids. So these are the key things: the structure and the properties. I've tried to keep myself focused by giving myself little titles. And here, if you're comparing and contrasting, I would always talk about one thing is like this and the other thing is like that, to make sure that you are making that clear contrast. And again, I have put in where I think I have got the individual marks. Slightly frustrated that I missed out this mark. That was an obvious one that I should have got. And here you're asked to describe how lactose is formed and where in a cell it would be attached to a polypeptide to form a glycoprotein. There was me packing the information in here because I wasn't sure where the marks were, and put in that it got put together in the Golgi apparatus because this is where modification and packaging of protein molecules occurs. So now what I've done is gone through all of those little notes that I made in the margin, and I've literally done a tally here of how many times I lost marks according to not reading the question, not using terminology, and lacking drawing conclusions. And you can see that my main issue here was because I was missing clues in the question, and in particular this whole thing about using the word explain to talk about the biology processes behind something. And so that is what I'm going to be keeping a lookout for as I do more exam papers, but also taking note of some of the terminology that I noticed that they wanted me to use. That thoracic cavity in the lung question, and making sure that I talked about internal intercostal muscles contracting. So this is how I analyse the paper and how I learn from it when I do future questions. I hope this has been useful. If it has, please do subscribe. Please share with your friends, and in particular for this video, I would really appreciate any comments that you have on it.